Dear everybody, dear all, I am very happy to have Professor Gary Powell here again, at least in the visual space here in Szeged. He will give the talk about COVID-19 vaccines. He already gave a similar talk in the beginning of January for the Institute of Biophysics of the Biological Research Center. And at that time, many people from the audience said that it would have been nice to have it recorded, but we didn't do it. And now this time I will record it. And it will be not exactly the same talk because things have happened since then, including that Gary got vaccinated. And he has first-hand experience about the vaccine. And also the other things surfaced on the topic. So this will be a different talk. Also, the audience is somewhat different because this talk was advertised for the regional committee of the Hungarian Academy, and I will share this document, the invitation document. As you see, it is a joint series of lectures by the BRC and the Szeged Regional Committee of the Hungarian Academy. And this is the second talk. And as I said, it will be recorded. By the way, I'm Tibor Páli, but maybe also you can see my name. It will be recorded and the talk will be available on the YouTube channel of the SZAB, of the Regional Committee of the Academy. I would like to tell a few words about Gary Powell, just very short. So he started research and teaching in 1996 as assistant professor of chemistry at University of Clemson. And after a very active research and teaching career, he also retired from the same university and became in 2006 and became Emeritus Professor of Genetics and Biochemistry. And his research areas include plant genetics and biochemistry and protein structure and function. And I would like to add that although he already retired, he is still very active in science. For instance, last year, he was selected as an I. Dwayne Eubanks Emeritus Fellow for Distinguished Service to the Clemson University Emeritus College. And with this, I give the screen and the microphone to Gary. Please give your presentation.
Thank you, Tibor. Good morning or afternoon, distinguished colleagues in Hungary and other places. I'm happy for the opportunity you've given me through the invitation of my friend, Dr. Tibor Páli, to talk to you for a few minutes about the development of vaccines against the virus causing the current pandemic. With international attention focused on combating this virus, we have a showcase for modern science, from molecular structure through cell biology and clinical science. So let me take you through some of these studies. I'm not starting on this first screen. There we go. Okay, here we go. All right, I got the first screen up now. Okay, what is COVID-19? It's a viral disease caused by SARS-CoV-2, which is severe acute respiratory syndrome, and it's the second occurrence of the infection of this virus. It's highly transmissible via aerosols from coughing, sneezing, or even loud talking or singing, as churchgoers found out fairly early on. It's a worldwide distribution pandemic. The overall death rate is about 0.65%, but the symptoms are widely variant and eclectic. Here's a partial list of symptoms that people experience. Probably one of the more interesting ones is the loss of taste or smell. But there's a whole variety of different things that can be caused by the virus. And there's an increased risk with age and people with underlying conditions such as diabetes, asthma, obesity. And it's not clear what the common link might be. The one suggestion is that perhaps we're dealing with a disease of the endothelial tissue rich in ACE2 receptors that would enable us to explain things like the loss of taste or smell and the various effects on respiratory and cardiac, and also the diarrhea. So that's a suggestion that you can pay attention to, ACE2 receptors. This is a bar graph showing the age versus death rate. And it's broken out by males and females. And it's clear from just inspecting this graph that the older you are, the higher your risk is. It's actually a lethal disease for people of increased age, old people like me. So that also explains the strategy of once you've treated the health workers and given them some measure of immunity, why then one goes after the most vulnerable in the population, starting with the older people and working your way down to less old people. The virus itself is shown in this electron micrograph. It's called a coronavirus because of the corona or halo surrounding the virus when you show a picture of this type. This is an artist's concept of the virus, showing the various structural components with the envelope protein, membrane protein, hemagglutinin, esterase, and the spike protein, all embedded in this lipid bilayer surrounding the nucleocapsid and the RNA. Indeed, it is an RNA virus, and in many ways, we're lucky that it's an RNA virus because RNA is a fragile molecule, relatively speaking. Organic solvents and soap and water can disrupt this lipid membrane, releasing the RNA and exposing it to the numerous nucleases that are in the environment. It's also susceptible to UV, so it doesn't persist on surfaces, particularly if it's exposed to sunlight. It's primarily transmitted, as I mentioned earlier, by aerosols coming from other humans. And so being a fairly fragile disease, it makes it a little easier to deal with than some of the DNA viruses that were, for example, on cruise ships, the novoviruses. The SARS-CoV-2 sequence was first reported in China to the NCBI, or the National Center for Biotechnological Information. And it was this sequence then that was immediately seized on by various people who were interested in making vaccines. Just knowing it was a coronavirus told people already that the primary way that it enters the cells is by binding to the ACE2 receptor. And the binding complex was reported at the end of March in the American Journal of Science, again by a Chinese group, showing just exactly what the interactions are at the surface between the virus spike protein and the ACE2 receptor. So this was actually between these two discoveries and reports, we actually had a strong basis for beginning to develop defenses against the virus, as the testing was based on the sequence, doing PCR as the primary way of detecting these viruses, and knowing that the way in which the virus enters the human cell by binding to these ACE2 receptors that gives us a way of blocking the virus entering the cell and gives us a rationale for creating vaccines against the virus. This diagram, please don't take too seriously. I know it's too small for most of you to make out the real details. I simply want to draw to your attention. It's a much more complex diagram than when I first gave this talk, because they've unsorted a lot of the functionalities of the non-structural proteins. This 29,903 base RNA is shown here with the various locus. The structural proteins are transcribed from base 21,555 into the S protein and the E, M, and N proteins that were described in the previous diagram. The other non-structural proteins are shown here in blue. There's some overlap because there is a frame shift, and that means that this transcript, as in many viruses, leads to the transcription of more proteins, more different proteins, more peptides than you would expect from just a linear transcription of the viral genome. Also be aware that there are proteases in the function that take the primary transcript and cut it up into polyproteins, and the trimming of these polyproteins also leads to difference in proteins. The primary functional things that one sees here, there are ways that the virus protects itself from the defenses. It blocks some of the apoptosis reactions, for example. It degrades host RNA. Another key feature that came from looking at the sequence is that there is a proofreading function built into the viral genome. It's not as elaborate as our proofreading functions, but there is some stability to the genome that arises because it does proofread its RNA transcripts, which arise from the RNA polymerase that's encoded by the virus. All right, let me go back now and review for you some things you probably already know, but let's put it up front so everybody's on the same page. The question is, what is an antigen and why are antibodies against viral antigens interesting? Antigens are molecules that initiate the immune response and can be bound by antibodies. Components of a virus alert the immune system to infection and trigger reactions that can result in clearing the virus and infected cells. B cells produce antibodies. T cells attack infected cells identified by bound antibodies. And the complement system enhances these specific immune responses. Vaccines are substances that trigger specific immune responses. A vaccine can produce immunity. While all 4 structural proteins of the virus are potential antigens, as are all 16 of the non-structural proteins, the spike protein is the most targeted antigen for the SARS-CoV-2 virus. Drug treatments may include inhibitors of the actions of any of the viral components that are required for reproduction and release of new virus. Inhibitors of the proteases, for example, are being looked at, as are inhibitors of many of the other 16 non-structural proteins. However, none of these drug candidates have proved effective. And so I state that there are a few treatments. There are a few treatments. One of the difficulties that one gets into is that when the virus invades, your immune system can overreact, and you can end up with a cytochrome storm, in which the immune system overreacts. And they found that dexamethasone, a rather inexpensive steroid, turns out to be one of the more effective treatments for people who are really sick and in this kind of situation. There are also a variety of monoclonal antibodies that have been developed and can be infused to block the virus. They're more effective when it occurs fairly early in the disease. And of course, that's difficult because if it's fairly early in the disease, you're probably not particularly sick. But anyway, there are these monoclonal antibodies that are available that can be used as treatment. And at the present, that seems to be the only real things that are effective. What are antibodies? Antibodies are large proteins produced by our bodies in response to something foreign, like a virus or a bacterium. An immunoglobulin is designed to bind the foreign body and attract immune components to destroy the foreign body. The two major types of antibodies are the IgM. An IgM is a pentamer of 220,000 Dalton proteins, and they're produced first. Eventually, they mature into IgGs, which are smaller, about 150,000 Daltons. And these IgG have the advantage of being remembered. There are memory cells that can store the information in that the IgG was successful in attacking the antigens brought by the virus. And if you get exposed to the virus at a later date, these IgGs can be produced rather quickly and present to help you resist the disease. We are said to be immune when this process occurs. Vaccines are designed to produce specific IgG and stimulate T cells. I might mention that, that these are families of molecules. There are literally tens of thousands of each of these, of varying specificity produced when you develop an immunity to a particular organism. So you have a whole arsenal available to you when you immunize yourself using vaccines or if you're infected. During discovery in animals and later phases in humans, these responses are an important measure of immunity and are closely examined. In the next slide, I'm going to show you a time course of the appearance of antibodies in humans. Again, it's a schematic, but if across the top, there's a window period before symptoms come on. There's a decline in the symptoms, if you're lucky, and a convalescence period that appears over a period of time. The days are shown at the bottom. Whoops. The days are shown at the bottom. So 0 would be the time at which you're first exposed to a virus. And then you gradually develop these symptoms. And it's shown here probably with some arbitrariness. 7 days, after 5 days, you see symptoms developing over a period of a couple of days. And then you begin to recover, and eventually after the end of a few weeks, while you're free of symptoms and you're quote normal. If you track the IgM, after about 3 days, IgM begins to be developed, and you can detect it coincident with the onset of the symptoms. And then it begins to decline as IgG production begins after about a week. IgG production is shown here as peaking around 20 days and gradually decreases after the infection has been cleared. If at a point someplace in here, after about 3 weeks, if you boost the immunity by another dose of antigen, either naturally or in the case of our vaccines, boost our immunity again by the vaccine, why this red line would take off again. And you would also favor the memory cells storing the information that has been generated by IgG, by these families of IgGs. All right, I'd like to now talk a little bit about Hungary. When I gave the talk, why I wasn't able to do this, but you have 5 vaccines available to you in Hungary as of the 21st of February. I'm going to, the slide shows you how many doses of the different vaccines are available. And what I propose to do is to talk to you briefly about a few of these vaccines and how they work. And I have a set of slides that are keyed to that. And then I'll develop the vaccines all together. By the way, the United States has Pfizer, the ones that have emergency use authorization, which is a special designation by our Federal Drug Administration, allows these things to be widely used, but no vaccine has been given full approval in the United States. So when one talks about approval, I'm going to use that in a generic sense, in the sense that here in the United States, why there's no full approval. But in some countries, why some of these vaccines have been given full approval on the strength of the preliminary data that they made available from their phase 3 trials. Anyway, with that proviso, we'll continue. Pfizer BioNTech has a vaccine that's based on RNA. BioNTech is a German company. Pfizer, of course, is an American company. And what one often finds is that a smaller company developed an idea. For example, Moderna, the last vaccine listed, is also an RNA virus, but they had no track record. They had never made any successful vaccines before. They were interested in cancer and were using RNA technology to try and treat cancers. And they've learned a lot. They had quite a bit of depth, some 10 years of experience working with RNA and trying to present it to human cells, but just had not tried to apply that kind of technology to developing a vaccine. Nonetheless, they were very quick about doing it. As soon as that sequence came out for the virus in early January, why they had already in a couple of days designed a vaccine based on their technology and what they knew about how to develop the RNA, and began putting it through the paces, the various stages that they needed to do that to test. BioNTech was doing the same thing. They had access to the same technology through patents which they'd purchased that actually they shared with Moderna. And so it's no surprise that they both were able to develop their vaccine with pretty similar time scale. You have 275,000 doses of a vaccine from a Chinese company, Sinopharm. You have 84,000 doses of the AstraZeneca vaccine, which comes from Great Britain. You have 23,000 doses of the Sputnik 5 vaccine developed by the company Gamaleya, that's part of the Russian government. All right, now what I propose is to move forward with a set of schematic slides taken from the New York Times that will give us some insight into how this works. Okay. Clearly, I'm going to talk about Sinopharm first. I do that because that's the simplest technology. The Beijing Institute of Biological Products created a vaccine, BBIBP, which was named with very little imagination based on the name of the institute. And the techniques also require very little imagination. They grew a whole lot of the SARS-CoV-2 virus and inactivated it. I use a little imagination. Imagine pounds of this stuff. You'd have to be very careful of it. And then when you inactivate it, one of the common inactivators is a beta lactone that modifies the DNA, and so makes it, and the protein as well, and makes it non-infective. And you'd have to be very careful that that was complete. Just a few virus particles in the vaccine that weren't completely inactivated would be a disaster, of course. But this technology has been around for a very long time, certainly tens of years. And there are vaccine making facilities available in a lot of countries. And the technology has been developed and thoroughly vetted. And so I think this is a good vaccine. If efficacy is 79.34%, and the trials in UAE provide even a higher efficacy rate. And the vaccine has been approved in a variety of countries, including Hungary at the present time. China, of course, is trying to use this as an instrument of their foreign policy. I might point out one more thing, being a vaccine based on inactivation of the intact virus, and given the fact that when you develop immunity, you do it to a whole variety of different antigens, that this virus probably, this vaccine probably provides a broad scope way of developing immunity. That is, it's not based on just one component, the spike protein. So this actually might prove to be valuable as we go forward. The next vaccine I want to talk about is Gamaleya. Gamaleya is developed by a division of the Russian Ministry of Health. And they called it Sputnik 5, that's Putin's term. They used it another non-imaginative name. It is a two-shot, two adenovirus vector vaccination that's based on delivering DNA that encodes the spike protein. It has an efficacy of 91.65%. And I've listed a recent publication that just came out this weekend, 20th February in Lancet. What happened was that they first used the vaccine on Army, on soldiers, and the results were good. But soldiers, of course, you're talking about young Russians, young Russian males of a fairly restricted section of ages. And when you want to test this thing use this thing on a variety of ages, well, you need to test it with old people and women and so on. So it wasn't really properly tested, but the Russians announced rather quickly that they had a vaccine that was very good based on their initial results, but it was met with some skepticism because of the reasons that I just outlined. They've gone through now and done a full-blown phase 3 trial using 19,866 volunteers over 18 years of age from a variety of age groups and ethnicities, presumably. The side effects were very few, and there were in a codier that large, you'd expect there to be a few deaths, but after investigating it, why they feel confident that the few deaths that were observed, 4 of them, were not linked to the vaccine. Because the DNA is encapsulated in a nucleocapsid, it's actually very stable, and it is a DNA after all, and you can store it at normal refrigeration temperatures. The vaccine has been approved in 27 countries, including Russia and Hungary. The schematic diagrams I'm going to show now show the basis for the virus. The DNA is shown in red here, encapsulated in this nucleocapsid of the virus. And the virus itself has been crippled. That is, it's not able to reproduce itself, not only because it doesn't contain viral RNA, at least it contains some viral RNA, but not the complete complement of viral RNA. There's been the RNA, and I used RNA. It's a DNA virus, and the DNA it contains some viral DNA, but not enough for that it can reproduce itself. What it reproduces is the DNA that encodes for the spike protein. The nucleocapsid is designed in such a way that it can bind to the surface of a cell and be engulfed. And then once it's engulfed, the virus is disassembled and translocates its DNA to the nucleus. And the nucleus the DNA is designed in such a way that it's rather quickly transcribed to RNA. The RNA is then using the machinery of the cell of which has been infected. This RNA now is processed and moved out into the cytoplasm, where it can act as a messenger. Before I leave this slide, let me suggest to you that if you have a RNA vaccine in which you have a piece of RNA that's encapsulated in a liposome, those liposomes can fuse with the surface of the cell and release the RNA directly into the cell. And so the rest of the things I'm going to be talking about are the kinds of things that the RNA virus, that the RNA vaccine would do. So the steps are going to be the same. Once in the cytoplasm of the cell, the messenger RNA is transcribed by a ribosome into protein of known protein sequence. That protein transcribed is then going to form a spike through, the spike is trimeric, and so these spikes are folded up and presented to the surface of the membrane and can be expressed on the surface of the membrane. And the immune cells, where the immune cells can find them and recognize them. There are some vaccines that are constructed of protein. That is, some companies have found a way to express these proteins quite efficiently, and then they package the protein up and present it as an adjuvant, as a vaccine. I've looked at that. It looks like a reasonable process if you present proteins, viral proteins, to our cells while there's going to be an immune response. But unfortunately, we just don't have much data yet about that particular process. I might add, after having some experiences in Derek Marsh's lab, as Laszlo and Jorg, we know that these membrane proteins are relatively intractable. They're not particularly soluble. They're difficult to deal with. And if you have spike proteins like these and you try and make large amounts of them and then put them together in some way to present them to the cell, that's going to require a fair bit of art and probably some things like detergents and various other kinds of tricks to properly present that to the cell. And it'll be very interesting to watch that and see how clever people be about presenting membrane proteins as vaccines. One trick that I've read about is the use of nanoparticles in which the protein is intermixed with nanoparticles, which present a large surface area. So it'll be interesting again to see how that works out in trials. Next, these proteins, either on the surface of a cell or released into the surroundings, are found by antigen-presenting cells, shown here by this irregular shape. They engulf these foreign particles and present portions of these viral particles on the surface of the cells where they're detected by a helper T cell. This process recruits other components from the immune system that can stimulate B cells and killer T cells. Shown again schematically, the helper T cell helps the B cell, which when it detects these particles on a vaccinated cell or an infected cell, begins to develop antibodies from the B cell. And the schematic is supposed to show the IgG, and the IgG then bind to the virus. They're specifically developed to bind specifically to these spike proteins, and by covering up the spike proteins, it's basically unable to infect. An antigen presenting cell showing these particles on the surface can also interact with a killer T cell and instruct it to find cells expressing spike proteins on the surface of the infected cell. And the killer T cell has, it's called a killer T cell because it can kill cells, and it can also recruit components of the complement system that are also lethal to the cell. And this is the way in which our immune system takes advantage of antigens, either from naturally from an infection or from the vaccine to result in immunity. In the case of the Sputnik 5, there's actually 2 doses, and the doses are different. There are 2 different adenoviruses, an Ad26 and an adenovirus 5. The first dose takes advantage of the adenovirus 26 containing a spike protein DNA, and the second dose is a different adenovirus. The idea is that when you infect cells with even these crippled adenoviruses, you probably make antigens against the capsid and the viral components. And if you then present the virus at a later date, 3 weeks later, if you've developed immunity to the virus and the virus, some of the viral components, the adenovirus, you can attenuate the effect of the second injection because you now have immunity against some of those components. And the thought is that by using a different adenovirus, you can circumscribe some of those effects. And it looks like it's effective. The efficiency and efficacy is actually very good. So Tibor, you're fine.
Okay, thank you.
Another vaccine that is available to you in Hungary is that made by AstraZeneca. Oxford University's Jenner Institute began working on a vaccine in January 2020. They used spike protein encoded in double-stranded DNA. Well, okay, they used a double DNA that encodes for spike protein in an engineered chimpanzee adenovirus. This chimpanzee adenovirus can't grow in humans. So that's by using one that, and it's not likely that we would have been exposed to a chimpanzee virus in normal everyday life. So it's for the same reason that I mentioned earlier, it's not likely you're going to have any natural immunity to these adenoviruses. And it is a matter of concern because the colds that we commonly get this time of year can arise from adenoviruses, so there can be natural immunity to these vectors that we're going to try and use to deliver the DNA, just as a matter of the way in which we live and the kinds of surroundings we have. But as I say, we're not likely we're going to see a chimpanzee adenovirus. They get the effectivity efficacy that's reported is 82.4%. They began phase 1, 2 in April. They formed a collaboration with AstraZeneca in phase 2 and 3 trials in May. The reason that an institute would form a relationship with a large drug company is that the drug company do these large trials all the time. And if it's not done right, well, you get into serious trouble. And so that's actually worth the money to involve these large companies so that you're sure it's done properly after you've done all the work, all the development work. In fact, this AstraZeneca is actually a textbook case of what the kinds of trouble you can get into. There was some confusion about the doses given during the clinical trials. Because of errors in the dilution of the vaccines, some subjects were given half the intended dosage. And surprise, the ones given the lower amounts, the efficacy was 90%. It was actually higher than the 62% that we got from the higher dosages. When they began looking at the data, trying to figure out what was going on, they also figured out that the lower dosages was given to 55 people, 55 years of age and younger. So you had two things going on. And when you start getting more than one variable as scientists, we know that gets you in trouble pretty quickly. So even with professionals handling these phases, the phase trial, the phase 3 trial, they got themselves in and I think they're just now unsorting it. I think the bottom line is that it's an acceptable vaccine. It does a good job, and it's approved in UK and 30 other countries, including Hungary. And I think they're saying that there's nothing wrong with that particular vaccine. Finally, the last two vaccines, Pfizer and BioNTech. It's a RNA vaccine, and this is brand new technology. We've never seen a vaccine made this way before. What people who have tried to inject raw RNA into the cells have found is that the RNA is an antigen. So if we're set up to attack viruses that enter our systems, and the attack is fairly severe. So if you just supply a raw RNA, it's going to be destroyed rather quickly. There are two ways in which the RNA is protected. The RNA that the patent information, the RNA has a modified 5' cap. Certain of the bases have been methylated, and some of them, for example, pseudouracil, are used to prevent it being an antigen, so it can dodge that bullet. You also need to enhance its ability to express the spike protein in human muscle cells. I learned that Katlin Kariko, who received her PhD from Szeged University and spent some time there at the Biophysics Institute in Szeged, and before she moved on to the University of Pennsylvania in the United States, is a world expert in this technology. It was her, when the university sold her patents, the patents were picked up by Moderna and used in their vaccine. So if you read about her in Wikipedia, you learn that she fairly quickly realized that she'd kind of sold the family home, and she still had some ideas and want to work on this some more. So what she did was she got herself hired by BioNTech and is now a vice president there. I said Pfizer. I'm actually not sure whether it's BioNTech or Pfizer, but anyway, she's still there.
Sorry, I think she's vice president of BioNTech.
BioNTech, okay, that makes sense because that's where the real science is taking place. This RNA that is specially protected is also enclosed in liposomes. The liposomes contain diasteryl phosphatidylcholine, cholesterol, and other lipids. I also learned that lipids can cause a certain soreness upon injection. So that's probably what I felt when with my first injection. As I mentioned, RNA is labile, so you don't have to worry about integrating into your genome, and you don't have to worry about it lasting very long if it doesn't find its way into a cell to be expressed. So it's probably very safe. And phase trials for the vaccine started August 3rd. The vaccine was the first COVID-19 vaccine to be given UA approval in the US on 11th of December. The vaccine is stored at very low temperature. Many of us have these deep freezers in the laboratory, so I'm a little bit surprised that was thought to be a big deal. There are recent reports that they have paid some attention to the formulation, and it can be stored at usual freezer temperatures now instead of having to do the deep freeze. This is the vaccine that I got vaccinated for on the 26th of January. I got my second vaccination on the 16th of February. The jab gave me a slight soreness, probably from the lipids. But I did have a reaction some 14 hours after the second vaccination when I experienced chills and shivering at night. But I think a bad night is not a bad exchange for the kind of immunity that I hope that we've achieved. Moderna is the second vaccine to receive just UA approval in the US just a week after Pfizer. It's also an mRNA vaccine, and I've said enough that you realize that they're very similar. I don't know what the RNA looks like. There may be some differences there, but and there might be some differences in the lipid composition of the liposomes. But it's the efficacy is just 0.5% less, probably within error. It was a new startup, so it was a big surprise that Moderna was able to develop a vaccine as quickly as they had. Pfizer had, for example, has had a lot of experience with making some of these vaccines. You know that Zika and Ebola, there were a lot of these countries, a lot of these vaccine manufacturers were paying attention to those, and so but they disappeared rather quickly before they really got a chance to fully test them. But now they have a chance to try out all this new technology and see how it works. A final note here, both these RNA vaccines are relatively easy to change. All you have to do is alter the RNA sequence and you have a vaccine that's new. So you can prepare something against some of these variants that we'll be talking about shortly. In principle, if the variants become a problem, we could go back for they could manufacture fairly short order, vaccine speaking, an altered vaccine with a slightly different sequence that might be effective against some of these variants. Also, if we have a whole new pandemic based on a totally different virus, again, all you'd have to do is change the sequence of the messenger RNA that's involved and administer it. Because the technology is tested now, you might expect that approval would be abbreviated. From what I read, with no direct evidence, Moderna may very well be involved in creating some vaccines that are specific for some of these variants at the moment. All right, so let's talk about these variants for a minute. That's come into the news just in fairly, again, it's very fast moving. SARS-CoV-2, as do many viruses with their rapid multiplication rates, undergo mutations, and thousands of variants have already been identified. The virus has proofreading functions, stabilizing its genome and slowing the mutation rate compared with something like influenza or AIDS virus, for which we have no vaccine. The AIDS virus we have no vaccine against. because it's so good at changing its surface. But this virus, if with the proofreading function, we would expect these vaccines to be potentially effective for months or years. Most mutations are point mutations with no functional consequences. Recall that some amino acids are encoded by up to 6 different codons. So that means you can change 6 different bases within the codon and still have the same amino acid. There is some use in doing the sequencing, however, because you can see that it becomes a name tag for that particular virus. So if you have a community where where you have some disease and you want to see if the disease transmits to another community, you can look at the sequence and see if the virus is similar, identical, or if there's major changes. And so get some sense about how it's transmitted. That plays into using testing and social distancing to control the virus. Some functional variation, particularly in the spike protein, does have consequences. Changing in the binding strength of the variants for ACT2 receptor seems to make it more infective, as we've seen in the UK, in Brazil, in South Africa, and as I read yesterday and today, some variants in California. Note that these changes in infectivity, that is the efficiency in which it's transmitted from one person to another. I mean, it's pretty fast the way it is, but apparently it's even better with some of these changes. It doesn't necessarily mean that an increase in infectivity means a change in disease severity or death rate. Those are separate properties. And you can test for those separately. So, and people have worked out ways of doing that. I read a paper yesterday where there are separate ways of testing for the binding strength and for working out the binding to vaccines and some of these monoclonal antibodies. And fortunately, you can change the spike protein and make it bind better to the ACT2 receptor, but you still expect a vaccine to produce immunity against that spike protein because you haven't changed the spike protein globally, just a few, maybe an amino acid change or two. So what we'd expect is that the vaccines may have reduced efficacy, but it should still work. So of course, as we reduce the number of active cases, these variants are going to be less of a problem. So I think the bottom line here is that we need to press on with the vaccination program, and it's kind of a race now, isn't it? We have to hope that we quench the viruses and reduce the number of cases before we develop a variant that eludes the vaccine, as seems to be the case for the AstraZeneca vaccine in South Africa. All right. You should be aware that the vaccines that we've talked about aren't the only vaccines going on. Again, out of the New York Times, there's something like 37 different vaccines that are in phase 1, 28 in phase 2, and 20 in phase 3. So there's a lot of vaccines out there, and they probably most of them are against the spike protein, but they're probably people starting to think about making it attacking other components of the virus. There are 6 and 4. I may have this EUA, it probably should be over here, but there's fully 10 vaccines that are approved and being used actively. I might also mention that just because you know a lot about vaccine structure and have good ideas and seem like they work, doesn't always work out that way. they list four vaccines that have been abandoned. I know that Merck, for example, had two underway, and the efficacy wasn't good enough. It simply didn't work very well. And Sanofi also has some vaccines that haven't worked. So it's not a home run. All right, I'd like to talk a little bit about the drug discovery process and give you some sense of what's been accomplished, because to generate working vaccines within a period of time that we've done has just been quite remarkable. This is a diagram that's designed for drugs, not necessarily vaccines. And this green is a series of processes that normally take place in laboratories, small laboratories, where people get ideas about how a drug might work, and then they try it out. And they usually use model systems of various kinds. If they have, there's a price tag with doing that kind of discovery, and the failure rate is actually pretty high. But eventually, people come up with things that can be tested more systematically on. It shows mice, rats, or dogs, and you again check this out. It normally takes a period of years and another chunk of money to work that process out. If you get something that really looks promising, you then think about moving it on to humans. The phase 1 trial, you have maybe 100 people, something like that, and you inject those people and watch them fairly carefully and see if there's any kind of reactions that you didn't anticipate. And again, that takes an indeterminate amount of time and a chunk of money. Phase 2 is proof of concept. You use a larger group of people, and you begin to look and see whether or not it actually does. This one was to see if there's something you didn't think of, the people getting sick. Here, you're going to actually start looking to see if it actually does what it's supposed to. In the case of vaccines, while you can assay for antibody formation, and that's a pretty good tool. And also, you can also have a look at your killer T cells and see if they are responsive. But eventually, you go to these large multi-center trials, and these are the ones that are really expensive because you have to pay them. So you're talking about a chunk of money, and it takes a number of years, again with a fairly high failure rate, before you again go on to licensing and full approval. Now, what has happened in the case of these vaccines is we were able to leapfrog past this early stuff because we knew that the coronavirus was going to need a spike protein to be infective. So you could skip these first stages and all the money associated with it to and step directly to the spike protein, as we've done, and begin working in phase 1 and phase 2. Very often these phases were combined. And these phase 3 trials were begun in many cases in summer, late summer, early fall for the vaccines. And again, it was fairly abbreviated. Normally the companies have to round up a chunk of money and pay these volunteers. And of course, something like half of your volunteers are going to get a placebo. The only real drawback is that unless you infect them with the virus, which is really unethical, you have to wait for them to get the disease naturally, and you have to wait for the placebo people to get sick or get the virus before you really have a proof of what's going on. So that actually took most of the time in these phase 3 trials. I say money wasn't an object. Here in the United States, we had a thing called Operation Warp Speed. It was a governmental private collaboration based on the CARES Act passed by Congress. And the previous administration selected companies and just put a lot of money on these companies and enabling them to scale up. Some of these companies stockpiled vaccines before they really knew that they were effective. And so it would be a bunch of money down the drain. And a company can't afford that, but a government apparently can. President Biden has closed this program and asked that the name no longer be used. The Federal Drug Agency allowed combining phases and used this early use authorization as a way of getting things up front. Also, so here's Johnson & Johnson, one of the first awards, $456 million award. Here's Sanofi, $31 million. And as I mentioned, their vaccine didn't work out. Here's Moderna. They got a really large award, even though they had no track record. AstraZeneca got a major award back at the end of May. So we should be seeing their vaccines here in the United States shortly. Johnson & Johnson, by the way, they keep telling us that we're going to see their vaccines momentarily. But it just seems like the weeks go on and we haven't heard anything more. I suspect there's something going on we don't know about. Notice that there is some other, we developed some new technology for the syringes. I think the second shot I got, the injections are fairly small. It's only something like 0.4 of a cc, which is a pretty small injection. And when the syringe is expelled, when all the contents of the syringe are expelled, the needle automatically retracts into the syringe, and then it can be disposed. So it makes disposal quite easy. Emergent Biosolutions, they got a major award. The growth media for these major companies, the various supplies that you need to grow cells to produce the adjuvants and the various things around doing this, they actually provided a supply chain for that. Corning developed a special kind of vial that's used that could be frozen at low temperature without a problem. Notice that there was some money for a drug that we now know doesn't work very well. Novavax is the company that's making a vaccine that's based on spike protein as the primary antigen. And Regeneron has an antibody that has been used in treatment. Now, I didn't try and update this. The numbers are not obvious. The previous administration wasn't very forthcoming about how they selected these things. So there's going to be some political byplay around this. All right, now, Tibor, shall I go on and talk about testing, or are we about done?
I think we are now at 55 minutes.
Okay.
So I suggest to accelerate and get closer to the conclusion.
I have a couple of slides about testing that's been interesting, because Clemson University has actually been very innovative about that. The standard test, you can see, involves a long wand, and people don't like that. It's expensive. You need special people to shove a wand like that down your nose, and it's time consuming. And once you pull that wand out, it's infective, and you have to have special plasticware to store it in. Well, this lady, this lady that's shown in the picture, Delphine Dean, said, well, why can't we do spit? So she started working in the early summer, developing technology using spit to do the testing. And guess what? It really works well. You don't need all that special hardware. Anybody can do it. The virus is very stable. It doesn't require special viral transport media. It's thick, so it actually didn't work too well until you heat it. If you heat it to 95 degrees for for half an hour, while it becomes much less thick because the polysaccharides and proteins in the spit degrade somewhat. But you and you release the RNA, but the RNA turns out to be quite stable in that kind of a milieu. So she got certification from the CDC, and we routinely test our athletes, and the staff and faculty all do this. The students get tested a couple of times a week, and if somebody turns up with COVID, why they quarantine them. They send them off to a dormitory where they for 7 days and test them, and when they're virus free, why then they're allowed to go back to class. We're doing our classes pretty much hybrid now. That is, some teachers meet with their students using suitable protocols, and then the rest of the time while they're doing it on Zoom, just like we're doing. So that's worked out for us very well in Clemson. We actually have a low infectivity rate, and it turned out to be a very useful kind of thing. The other thing that might be worth noting is that Duodna has found a way of doing really, really fast PCR tests. She uses an agent, CRISPR-Cas13, that specifically binds unamplified CoV2 RNA. And when it specifically binds this, and you say specifically, you use a piece of RNA that it codes for CoV2, and to instruct the Cas13. And when the Cas13A is instructed, then it develops ribonuclease activity when it encounters that piece of RNA. They use a probe. It's a fluorophore separated from a quencher by a short piece of RNA. And when the Cas13A is activated, it cleaves the RNA and the fluorescence interacts. It means that you can do the assay in 5 minutes using an iPhone. So I thought that was pretty cool. So testing will get even better, and all right, here's the summary slide. We've got vaccines available. The state of South Carolina, population 5.1 million. It's a little bit more than half as big as Hungary. So it's actually a reasonable sort of thing, model for Hungary. They've vaccinated almost, as of today, they had vaccinated almost 600,000 people. We're using the Pfizer and the Moderna. I reiterate how that virus is fragile. Folks, we're really so lucky that this thing has its properties. It's being a fragile virus. It's proofreading, so that's going to help us. There are mutants and variants, but we're going to learn to counter those mutants and variants because we have this RNA-based vaccination that is really facile and will help us so much as we go on. All right, Tibor, if you have any questions, I'd be glad to take them.
Thank you very much. This was really a different talk from the first one. I'm surprised that so many things have changed since then. And it was very clear, and I enjoyed very much. And if anybody has a question, please raise your hand or turn on your microphone. Are there any questions? Laszlo Zimányi has a question, and also Derek. So Laszlo first, and then Derek Marsh.
Okay, well, Derek should be the first, but I'm trying to copy something to the chat. Yes, I did. I just want to call your attention, and also mostly the non-Hungarian participants' attention, to another piece of news, that there is a small company in a small village in Hungary, and the lady who operates this company also worked for a while in the BRC, and this company is the one that provides the anti-double-stranded RNA monoclonals to all these RNA vaccination companies. This is if you check out the link that I sent to the chat, it's worth trying to check out. And it's another little piece of information, another contribution by a small Hungarian company to this whole story. All these mRNA vaccine producing companies, well, not only them, many other companies use their double-stranded DNA. And I'm also, can I also attach a file, Tibor, or what do you think, or a file not?
Sorry, to share, you can share.
Oh, so share a file. How can I share? Because I have a picture of this village, this small company. How can I share?
Share screen, and then you select the right window.
Share the screen. Well, share the screen.
Bottom, green.
Shall I? Just to show you. So can you see this picture here?
Yes.
So this is a beautiful little village in a hilly part of Hungary, and there is an old peasant's house, which is the house of the great-grandmother of this scientist lady, and that's where, you don't see the house, that's where they produce a double-stranded DNA. Okay, so that's all I wanted to say. Check it out. Okay, I stopped sharing if I can. Yes, stop there. Interesting connection.
Derek, you have a question. Please turn on your microphone.
I'm here. Thank you, Gary. My first question is about the expression of the spike protein. You implied, at least in the DNA case, that it expressed the whole protein, including the membrane anchor, whereas from the EMs, one thinks that the majority of the spike protein and its antigenic parts will actually be soluble protein. I mean, is actually membrane protein expressed, or just the soluble external part?
The short answer is I really don't know.
Oh.
I know it's a baculovirus expression system, the insect, the moth, and that's really about all I know. You only see a certain.
No, I meant the design of the DNA in the case of the Russian one, and the messenger RNA.
Yes.
Does that sequencing include the transmembrane encoding part of the spike protein?
It's a really good question, and I'd love to know the answer.
Oh. My other question, which was the one that I was going to ask last time, if I'd have been able to get it to your seminar soon enough, was, I mean, this business, you mentioned liposomes, and it's very nice because we've worked on liposomes for so long, but my understanding is that, in fact, they're not liposomes as we know them, i.e. a bilayer vesicle, but they're these so-called lipid nanoparticles, and that doesn't explain what it is, but it seems to me that I'm out of it, but this is part of the jargon is that are specifically lipids, including positively charged lipids, specifically for encapsulation of nucleic acids.
Okay.
So I might, well, Tibor, in fact, looked up the Pfizer lipid composition, and there was something that, I mean, well, you could probably, you'd read the name better than I, but it looked as if it was probably a positively charged lipid that was one of the constituents of the lipid mixture. I mean, there was cholesterol, there was DOP. I think it was DOPC, in fact, not DSPC.
Okay.
And two other components with a systematic chemical name, of which one of which I think was a substituted amine function, which would be positively charged.
Okay.
Because I just wondered if you'd followed that part of it, because it seemed I'd be interested to know how much is known about the structure of those sort of these so-called lipid nanoparticles. It could be an interesting area for biophysical research.
Yeah, if you think about it, what the idea of injecting these lipid nanoparticles and expecting them to fuse spontaneously with the membrane seems kind of interesting. There's a lot of biophysics, I think, that we aren't party to, and it would be very interesting, as you say, to learn more about that.
Yeah, well, the question is whether, for instance, if it's it's a bit like the inverse of cardiolipin, I guess. If you have a positively charged lipid binding to the phosphates of the nucleotides of the nucleic acids, you could have a sort of inverted hexagonal type phase structure, or at least an inverted micellus structure, which may in fact be more fusogenic than the normal circulating liposomes that were used for delivering soluble drugs.
Yes, that's really interesting, Derek.
But, the other question that I've been asking myself is, in the PCR assays, after you've done PCR, what is it that is then detected? Because it's, where is the specificity? Because we know from the new variants that in most cases, I mean, the test doesn't normally involve a full sequencing of the nucleic acid, does it?
No, and that's actually interesting. It's a variant on that slide that I showed where you have a quencher and a fluorophore.
Yeah.
Apparently, when you use the polymerase, one of the things it does when it gets to the end of the transcript, it actually cleaves that reporter group. So you see an increase in fluorescence. Mm-hmm. And so that's all you measure. Yeah, you let it run for a while and then look and see if you have an increase in fluorescence.
Thank you.
Mm-hmm.
Thank you very much. I don't see additional arms raised. Is there any other question? So there are no questions. Thank you very much, Gary Powell, again, for the nice talk, and also for the audience. I will now stop the recording, and we can have a free discussion if you are interested.